So dear friends, once again it's very hearty and warm welcome. Dear friends, all of you are known. In our previous lecture, we have already started the study of this chapter and this topic, that is Mendel's laws of inheritance. And in previous lecture, we have completed the study of first law of inheritance, that is law of dominance, as well as second law, that is law of segregation, along with suitable examples. Now today, first of all, we are going to study about Mendel's third law of inheritance, that is law of independent assortment. And then we shall study about back cross and test cross. So first of all, let us start the study of new topic, that is Mendel's third law of inheritance, that is law of independent assortment. First of all, let me tell you the statement of this law. Listen carefully. Actually, this law can be explained with the help of dihybrid cross, about which we shall discuss after 2 minutes. Let me tell you the statement of law of independent assortment. This law states that when pair of alleles controlling different characters, located in the different pair of homologous chromosomes. Listen once again. This law states that pair of alleles controlling different characters located in the different pair of homologous chromosome assort independently during gametogenesis, that is during gamete formation, as well as during fertilization, that is during coming together in zygote. I am going to repeat the statement once again, please. I said that law of independent assortment states that the pair of alleles controlling different characters, and located in the different pair of homologous chromosome, are independent of one another during segregation, during separation, at the time of gametogenesis, at the time of gamete formation, as well as we can say during fertilization, that is during we can say zygote formation and in the new offspring. This law can be stated by some another way. हा जो लॉ आहे तो वेगळ्या पद्धतीने पण आपण स्टेट करू शकतो. I am going to tell you. Listen. Law of independent assortment states that when two pure, that is homozygous parent individuals, differ in two or more than two pairs of contrasting characters are crossed. When two pure, that is homozygous parent individuals differ in two or more than two pairs of contrasting characters are crossed, then inheritance of one character is independent of the other character. Inheritance of one character is independent of the other character. Means, I want to tell you that the law of independent assortment, it can be stated by different ways. Here I am going to write first of all the statement of law of independent assortment at the blackboard. Listen. Law of independent assortment states that the pairs of alleles, the pairs of alleles controlling different characters located in the different pairs of homologous chromosomes. Different pairs of homologous chromosomes are independent of one another, during segregation. We can say during gamete formation, that is gametogenesis, gamete formation, as well as formation of zygote by fertilization. It means simply we can say law of independent assortment states that the inheritance transmission of one character into the offspring is not dependent on another character. It means one character permanently does not remain associated with other character. Now, I said few minutes before that this law of independent assortment can be explained with the help of dihybrid cross. We have already studied about dihybrid cross in our previous lectures, and therefore let us consider the same example. And before going to that example, just read this law of independent assortment once again. This law states that the pairs of alleles, it means either two or more pairs of alleles controlling different characters located in the different pairs of homologous chromosomes are independent of one another during segregation, during gamete formation, as well as formation of zygote. मराठीतून जर सांगायचं झालं तर आपण असं म्हणू शकतो, ज्या वेळेला एखाद्या सजीवामधले दोन किंवा दोनापेक्षा अधिक गुणधर्म एका पिढीमधून पुढच्या पिढीमध्ये संक्रमित होतात, त्यावेळेला एका गुणधर्माचं, एका कॅरेक्टरचं पुढच्या पिढीमध्ये होणारं संक्रमण हे दुसऱ्या कॅरेक्टरशी संबंधित नसतं. म्हणजे समजा दोन कुठले तरी कॅरेक्टर्स हे पॅरेंट मध्ये आढळत आहेत, पण पुढच्या पिढीमध्ये ते दोन कॅरेक्टर्स, ते दोन गुणधर्म पुढच्या पिढीमध्ये बरोबरच येतील असं नाही. याचा अर्थ एक गुणधर्म आणि जो दुसरा गुणधर्म आहे दे असॉर्ट इंडिपेंडेंटली, देअर इनहेरिटन्स टेक्स प्लेस इंडिपेंडेंटली. म्हणजे एका गुणधर्माचं पुढच्या पिढीमध्ये संक्रमण होत असताना त्याचं संक्रमण, त्याचं इनहेरिटन्स, त्याचं ट्रान्समिशन हे दुसऱ्या कॅरेक्टरवर डिपेंड नसतं. दे असॉर्ट इंडिपेंडेंटली, दे इनहेरिट इंडिपेंडेंटली, and that we can understand by considering the example of dihybrid cross. Now we have already considered one example, that is of two characters, such as during which we have considered two characters such as the shape of seed and color of seed. Now Mendel crossed two pure pea plants by considering two characters such as the color of seed and shape of seed. So when Mendel crossed two pure pea plants by considering two characters, color of seed and shape of seed, we know that the contrasting traits of color of seed are yellow and green, while the contrasting traits of shape of seed are round and wrinkled. Also we are known that capital Y stands for yellow color, small y stands for green color, capital R stands for round shape and small r stands for wrinkled shape. Therefore one parent is homozygous for yellow round and second parent homozygous for green wrinkled. Therefore, obviously, the genotype of one parent, one in the sense we can say pure yellow round, is capital Y, capital Y, capital R, isn't it? Because these parents are pure homozygous. It means in each allelic pair both the genes are identical. अर्थात हे जे parents आहेत ते प्युअर आहेत, होमोझायगस आहे. म्हणजे काय की प्रत्येक गुणधर्म नियंत्रित करणाऱ्या जीन्सच्या जोडीमधले दोन्ही जीन्स हे सारखे आहेत, आयडेंटिकल आहेत. मग येलो साठी या ठिकाणी असणारे कॅपिटल वाय, राऊंड साठी असणारे कॅपिटल आर. अँड अबाउट अनदर पॅरेंट दॅट इज प्युअर ग्रीन wrinkled. Genotype small y, small r. Actually, it is double homozygous condition. These individuals are called double homozygous because here both allelic pairs, both gene pairs show homozygosity. Anyway, already we have discussed while studying the dihybrid cross. Now, when these two pure parent individual crossed, we know they produces F1 individual, that is with genotype capital Y, small y and capital R, small r. We have already studied, and it has the phenotype that is yellow and round. Now here you can notice all F1 individual produced in this cross appeared yellow round. Okay. Now when these F1 individuals are self-crossed, that is capital Y, capital R, small r is crossed with same, that is capital Y, capital R, small r. ज्या वेळेला या F1 इंडिव्हिज्युअलचं सेल्फिंग केलं जातं, सेल्फ क्रॉसिंग केलं जातं, ऑब्वियसली टू ऑब्टेन द F2 जनरेशन. अर्थात F2 जनरेशन मिळवण्यासाठी, F2 जनरेशनचे इंडिव्हिज्युअल्स मिळवण्यासाठी F1 चं क्रॉसिंग केलं जातं, सेल्फ क्रॉसिंग केलं जातं. Now during self crossing we know as usual initially it occurs the gametogenesis. त्या ठिकाणी गॅमेट्स तयार होतील. Now listen, it is important. When this F1 individual undergoes gametogenesis, gamete formation, look, it has two pairs of alleles, isn't it? It has two pairs of alleles according to the statement of this law. Pairs of alleles controlling different characters. So here this F1 individual has two pairs of allele. One pair capital Y, small y. Another pair capital R, small r. So now initially during gamete formation what happens? These alleles they separate from each other. Alleles separate from each other. It means these two genes in this pair they separate from each other. In the same manner the pair of genes, these two alleles also separate from each other in this manner. Is that clear? And now after that what happens? One of the capital Y or small y they enter along with capital R or small r into single gamete. Try to understand. There is equal chance. We can say along with this capital Y, capital R can enter into single gamete, or we can say along with this small y, capital R can enter into one gamete. अर्थात या कॅपिटल Y बरोबर या दुसऱ्या जीन्सच्या जोडीपैकी हा एलेल येऊ शकतो, किंवा स्मॉल y बरोबर हा एलेल येऊ शकतो म्हणजे कॅपिटल R, किंवा पुढे आपण सांगू शकतो की कॅपिटल R आणि स्मॉल r या दोघांपैकी कोणीही या कॅपिटल Y आणि स्मॉल y बरोबर येऊ शकतो ड्यूरिंग गॅमेट फॉर्मेशन. मीन्स व्हॉट हॅपन्ड हियर? Actually, initially the alleles of each pair, they get separated from each other. They get segregated from each other. What we have studied in second law of segregation. Okay. And now during gamete formation and inheritance, one gene of each allelic pair can come with another gene of another allelic pair. Means capital Y can come with capital R. In the same manner, capital Y can come along with small r, or there is a chance the small y can come along with capital R, and another and last possibility, small y can come along with small r. It means when these F1 individuals undergo gametogenesis, at that time 4 different types of gametes are formed. Is that clear? 4 different types of gametes are formed. And why? That I told you just now. Now the 4 gametes of one parent and 4 gametes of another parent, when undergo fertilization, there is a random mating, and hence in all 16 individuals are formed in F2 generation. पर्यायाने या ठिकाणी 4 आणि 4 दोन्ही पॅरेंट्सचे 4 गॅमेट्स ज्या वेळेला त्यांचा रँडम मीटिंग होईल तर अर्थातच 4 into 4 in all 16 offsprings are possible in F2 generation. Okay, and we have already studied in dihybrid cross, and that 16 offspring produced in F2 generation, the F2 generation मध्ये जे काही 16 offsprings तयार होणार आहेत त्यांचं जीनोटाइप आणि त्यांच्यामधला रेशो हा आपल्याला माहिती आहे. We know that the F2 individuals during this dihybrid cross is 9s to 3s to 3s to 1, and also we know what should be the phenotype of these F2 individuals. हे जे F2 individuals आहे त्यांचं phenotype काय आहे हे आपल्या सगळ्यांना माहिती आहे. काय असतं हे आपल्याला माहिती आहे कारण आपण dihybrid cross मध्ये ते शिकलोय. Out of these 4 different types of F2 individuals, the 9 individuals with yellow and round phenotype, 3 individuals with yellow and wrinkled phenotype, next 3 with green and round phenotype, and 1 with green and wrinkled phenotype. Now please try to understand. I said that due to the random mating of 4 gametes of both of the parent individuals, say now here F1 individuals, in all 16 offsprings are produced in F2 generation, and they show as 9s to 3s to 3s to 1. You know, it is also called as the dihybrid ratio, isn't it? Phenotypic dihybrid ratio we have already studied. And here, out of these 4 different types of offsprings in F2 generation, 9 individuals appeared yellow round, 3 yellow wrinkled, 3 green round, and 1 green wrinkled. Now, out of these 4 different types of combinations, just try to understand, out of these 4 different types of combinations, 2 combinations are parental. That is, 2 combinations are similar to that of parent. Which are those? Look, yellow round, exactly similar to the parent generation. One of these 2 parents, yellow round. Then last, green wrinkled. It is also parental combination, just like another parent, green wrinkled. याचा अर्थ हे जे 4 वेगवेगळ्या प्रकारचे ऑफस्प्रिंग्स F2 जनरेशन मध्ये निर्माण झाले आहेत त्यापैकी 2 कॉम्बिनेशन्स हे पॅरेंट प्रमाणेच दिसतात, पॅरेंट्स प्रमाणेच दिसतात, येलो राउंड आणि ग्रीन wrinkled. But these 2 combinations are new. These 2 combinations or these 2 phenotypes are not seen in parents as well as we can in F1. Which are those new combinations? Yellow wrinkled and green round. What does it mean? it indicates that the yellow color of seeds is with round shape of seed in parent, or green color of seed and wrinkled shape of seed are also togetherly in parent, but during their inheritance, during their transmission, the yellow colored appeared with wrinkled. Yellow colored appeared with wrinkled and green colored appeared with round. It means the inheritance of one character or inheritance of one gene is not depend upon other character or other gene. You might have understood. Let me tell you. अर्थात या ठिकाणी एकूण जे 4 वेगवेगळ्या प्रकारचे कॉम्बिनेशन्स किंवा फिनोटाइप मिळाले आहेत F2 जनरेशन मध्ये, त्यापैकी येलो राउंड आणि ग्रीन रिंकल्ड हे पॅरेंट्स प्रमाणे मिळालेले कॉम्बिनेशन्स आहेत आणि 2 न्यू या ठिकाणी आपल्याला कॉम्बिनेशन्स मिळतात. याच्यावरून काय सिद्ध होतं की पॅरेंट्स मध्ये प्रत्यक्ष येलो कलर आणि राउंड शेप हे बरोबर आहेत, दुसऱ्या पॅरेंट मध्ये ग्रीन कलर आणि रिंकल्ड हे बरोबर आहेत, पण पुढच्या पिढीमध्ये संक्रमण होत असताना, पुढच्या पिढीमध्ये इनहेरिटन्स ट्रान्समिशन होत असताना येलो कलर हा रिंकल्ड बरोबर आपल्याला आढळतोय आणि ग्रीन कलर राउंड बरोबर आढळतोय. याचा अर्थ काय होतो की एका गुणधर्माचं पुढच्या पिढीमध्ये होणारं संक्रमण हे दुसऱ्या गुणधर्मावर डिपेंड नसतं. दे असॉर्ट इंडिपेंडेंटली. सो द नेम इज law of independent assortment. So here we have studied about the law of independent assortment. Now we have to study about the second topic, that is test cross and back cross. So now let us talk about test cross and then we shall discuss about the back cross. Let me tell you the statement. What does it mean? Test cross. Test cross, it is the genetic cross. Test cross, it is the genetic cross between an organism of unknown genotype with a recessive individual, with a recessive organism. Listen once again. Very simple. Test cross means it is the genetic cross between an organism with unknown genotype. Unknown genotype means the organism whose genotype is not known to us, with a recessive homozygous organism. It is the crossing between organism of unknown genotype with homozygous recessive organism. Let me write that statement here. Test cross, it is the crossing between an organism of unknown genotype. अर्थात ज्या सजीवाचं आपल्याला जीनोटाइप निश्चित माहित नाहीये अशा सजीवाचं रिसेसिव्ह होमोझायगस ऑर्गॅनिझमशी क्रॉसिंग याला म्हटलं जातं टेस्ट क्रॉस. आता उदाहरण घेऊन आपण ते समजून घेणार आहे. बट बिफोर टू दॅट लेट मी टेल यू व्हाय दिस क्रॉस is known as test cross. Actually, name itself indicates test cross. This cross is performed to know the genotype of that organism, of any particular organism. Look here, I have written it is the crossing between an organism of unknown genotype. अर्थात ज्या सजीवाचं ज्या ऑर्गॅनिझमचं जेनेटिक कॉन्स्टिट्यूशन काय, जीनोटाइप काय हे आपल्याला माहित नाहीये आणि ते समजून घेण्यासाठी, ते तपासण्यासाठी, ते टेस्ट करण्यासाठी या क्रॉसचा वापर केला जातो, म्हणून याला टेस्ट क्रॉस म्हणतात. ऑफ कोर्स दिस मेथड ऑफ टेस्ट क्रॉस was also devised by Mendel and still it is under, we can say, implementation and it is used by different plant breeders also, we can say animal breeders also. Anyway, so let me explain you by giving suitable example. Consider, suppose we have a tall pea plant, but we don't know what is the genotype of this tall pea plant, because you know the genotype of this tall pea plant may be either capital TT or we can say capital T small t. Phenotypically that plant is tall, externally that plant is tall, okay, but we don't know whether that tall pea plant is homozygous tall or heterozygous tall. Is that clear? Now to know its genotype, this tall pea plant is crossed with a recessive organism, recessive pea plant. Now as far as concerned with character, height of pea plant, we know the recessive character is dwarf. So if this organism, that is in concerned example, we can say tall pea plant crossed with pure dwarf pea plant, then this cross is nothing but the test cross. Listen once again. Test cross, it is the crossing between an organism of unknown genotype with recessive organism, and I already told you the purpose of this test cross is to know about the genotype of that any particular organism of unknown genotype. Now when here the tall pea plant of unknown genotype crossed with pure dwarf, then whatever ratio is obtained in F1 generation is if 1:1. That is, the F1 individual produced by the crossing of these two individuals of these two pea plants showing 50% tall and 50% dwarf. That is, if the ratio among F1 individual is 1:1, then it indicates that this tall pea plant is definitely heterozygous. It is heterozygous. Why this is so and how this is so? Let me explain you. We know that the dwarf pea plant always has genotype small t. Now if this dwarf pea plant, that is a recessive organism, has two alleles, recessive alleles, means definitely one allele has came from one parent, another allele has came from another parent. So obviously this tall pea plant has at least one small allele, that is recessive allele. So it indicates clearly this tall pea plant has a genotype capital T and small t. And if all the individual produced in F1 generation show only dominant character, say in this example tallness, then this tall pea plant is considered with genotype capital T. That's all about the meaning of test cross. Actually the test cross, it is of two types. टेस्ट क्रॉसचे दोन प्रकार पाडले जातात, अर्थात मोनोहायब्रिड टेस्ट क्रॉस आणि डायहायब्रिड टेस्ट क्रॉस. Very simple. Monohybrid test cross means what? It is the test cross in which only inheritance of single character is considered, or in this test cross only a single character is considered. While dihybrid test cross means it is the test cross in which two characters are considered simultaneously. Now, the ratio obtained during monohybrid test cross in F1 individuals is called monohybrid test cross ratio. अर्थात या ठिकाणी मोनोहायब्रिड टेस्ट क्रॉस करत असताना F1 इंडिव्हिज्युअल्स मध्ये जो रेशो आपल्याला आढळून येईल त्याला म्हटलं जातं मोनोहायब्रिड टेस्ट क्रॉस रेशो. आणि जर तो मोनोहायब्रिड टेस्ट क्रॉस रेशो, ॲज वी डिस्कस अर्लियर, तो मोनोहायब्रिड टेस्ट क्रॉस रेशो जर 1:1 आला तो obviously the organism with unknown genotype is heterozygous. Okay, in the same manner the dihybrid test cross is the test cross in which two characters are considered, and during dihybrid test cross, if the F1 individuals show the ratio as 1:1:1:1, of course it is known as dihybrid test cross ratio, then it indicates an organism with unknown genotype is double heterozygous. Double heterozygous means here simply we have to understand meaning of test cross and what is the importance of test cross. After the study of test cross, one more important term here we have to study, that is back cross. Back cross, it is the genetic cross. It is between hybrid individual or heterozygous individual. Hybrid. More precisely, F1 hybrid. F1 hybrid or heterozygous individual with any one of the parents. Any one of the parents. Any one. Let me give very simple example. When the pure tall pea plant, as usual, is crossed with pure dwarf pea plant. When pure tall pea plant is crossed with pure dwarf pea plant, then we know capital TT, small tt, that is genotype, gametes, and the F1 individual produced with genotype capital T small t and phenotype tall. Now when this F1 hybrid, that is tall pea plant, is crossed back with any one of the parents, हा जो हायब्रिड इंडिव्हिज्युअल आहे, F1 इंडिव्हिज्युअल आहे, त्याचं जर त्याच्याच दोन पॅरेंट्स पैकी कोणाशी तरी क्रॉसिंग केलं तर त्याला म्हटलं जातं बॅक क्रॉस. नावच आपल्याला सांगून जातं. बॅक याचा अर्थ रिटर्न. मग या ठिकाणी हा जो F1 आहे, समजा कॅपिटल टी स्मॉल टी, या कॅपिटल टी स्मॉल टी चं जर प्युअर टॉल या पॅरेंटशी क्रॉसिंग केलं तर हा पण बॅक क्रॉस होऊ शकतो. किंवा इफ दिस F1 individual is crossed back with this plant, that is pure dwarf small tt, it is also back cross. But we have already studied the crossing of any organism with recessive individual is known as the test cross. It means actually this type of back cross is an example of test cross. So we can say that test cross, it is also back cross, but back cross is not always test cross, isn't it? याचा अर्थ टेस्ट क्रॉस हा बॅक क्रॉसच आहे, एक प्रकारचा बॅक क्रॉसच आहे. कारण टेस्ट क्रॉस मध्ये आपण वर बघितलं की टेस्ट क्रॉस मध्ये एखाद्या इंडिव्हिज्युअलचं ज्याचं जिनोटाइप आपल्याला माहित नाहीये त्याचं रिसेसिव्ह पॅरेंटशी क्रॉसिंग केलं जातं. मग या उदाहरणामध्ये आपल्याला कळतंय की हा जो F1 इंडिव्हिज्युअल आहे त्याचं रिसेसिव्ह पॅरेंटशी क्रॉस केलं तर हा टेस्ट क्रॉसच झाला. म्हणून मी म्हटलं की टेस्ट क्रॉस इट इज ऍक्च्युअली अ बॅक क्रॉस, बट बॅक क्रॉस इज नॉट ऑलवेज टेस्ट क्रॉस. प्रत्येक बॅक क्रॉस हा टेस्ट क्रॉस असू शकत नाही. नाउ व्हॉट इज द सिग्निफिकन्स ऑफ दिस बॅक क्रॉस? या बॅक क्रॉसचं महत्व काय आपण समजून घेऊ. ऍक्च्युअली इन मॉडर्न जेनेटिक्स ऑल्सो द प्लांट ब्रीडर्स, एनिमल ब्रीडर्स, they perform the back cross to obtain the pure individuals quickly. To obtain pure or homozygous individuals of desired stock, कुठल्या तरी desired character असलेले इंडिव्हिज्युअल्सला होमोझायगस किंवा प्युअर कंडिशन मध्ये मिळवण्यासाठी ह्या बॅक क्रॉसचा वापर केला जात असतो. एनिवे, सो हियर वी हॅव डिस्कस्ड about the two topics in this lecture. That is, first of all, law of independent assortment, and then we have discussed about the test cross and back cross. So now today let us stop here. Thank you. Welcome.